Greetings and welcome to Meet a CHM Researcher. My name is Cory MacDonald. I am the Patient Engagement Director for the Choroideremia Research Foundation. And with us today is a absolute legend in the world of vision research, not just for CHM, but other eye diseases. And I'd like to welcome Dr. Jean Bennett. How are you today, Dr. Bennett?
Good morning, Cory. It's great to see you.
Same here. It's been way too long.
Yep.
This darn COVID thing is just killing my social life. So
yeah. Well, hopefully the end of this particular infection is in sight.
You know, it's funny. We normally have our conference every 2 years, and we had to, push back our 20th anniversary conference to 2022. And I never realized how much those conferences just energized me until we didn't have one.
Yeah.
And now I'm like, oh, I can't wait for this year. I need to recharge my batteries.
I know, we need a fix.
Exactly. So now, you live in Pennsylvania,
right?
But you also have a little place up in Western Mass as well. And so I love the fact when you sent me some pictures, with the famous PhD researcher up on her John Deere. I just said, this is perfect.
That's my favorite toy.
Well, it's better than your husband with a chainsaw. So
yeah. No, we have a great picture of him using the chainsaw with our daughter, who was then in diapers, standing right next to him, and he's sawing up logs.
Oh, that's beautiful. So what do you like to do when you head off from Philly and head up to Massachusetts at your little place there?
The place in Massachusetts is actually very rustic. So we spend a lot of time taking care of it. The heating system of the house is all through firewood. So we spend some time splitting logs, hence the chainsaw. And my favorite thing is to be a summer farmer. I love planting crops and harvesting them, and we raise bees there. We collect honey, and I just love being out in the middle of nature, having animals, walk upon us. We have turkeys, we have porcupines, plenty of deer, bears, etc. And it's such a great escape from city life.
I imagine, yeah, and I think Karen's going to have to consult with you on those pumpkins, because she does a lot of backyard gardening and grows, she has her cucumbers and blueberry bushes and things like that, but tomatoes, lots of tomatoes. But I know she tried watermelon and it didn't take.
Yeah, I guess the hard part is figuring out what will take in your particular land and climate. And I'm still learning.
Yeah. Yep. And the beekeeping. We'll have, I still laugh about that, the picture that Al posted on Facebook when he got stung on his tongue.
I got stung on my tongue. We were eating dinner outside, and it was dark, and it was Mexican food. And I go, that was really hot. In fact, that was really hot, and my tongue started swelling. Scary.
Oh my gosh. Oops.
Yep.
But hey, the honey is worth it, though. That's
totally. Yeah. Al's already gotten his first sting of the year because he can't resist checking on his bees, even when it's 30 degrees outside.
Yeah, there was that other picture he posted once about he had the thermal camera.
Yes.
A picture of the hive. I thought that was the coolest thing.
Isn't that great? Yeah.
And how hot it was inside that hive.
And how they cluster together. It's pretty interesting.
I see, that was really cool.
Yes.
It's a fascinating science. So, and then you also enjoy music. Now, now you play the piano. Now,
Yes.
wasn't your mother a musician as well? Did you tell me that once?
My brother was.
Okay.
He was a really famous oboist. Unfortunately, he died a number of years ago, but he was a professional musician. And my grandmother, my maternal grandmother, was a concert pianist who played
Okay, that's who it is.
on
Yeah.
New York radio, WQXR.
And so you
I have
play as
her
well.
piano. It's super special. I'm nowhere as good as her, but I love it. It's my religion.
Yeah, I mean, there's something very, with me, it's, singing, and whether I sing bad or good, I don't care, but I sing in it. That's kind of my thing. It's
I
what
can
helps to
relate.
relax me. But
Yeah.
I thought that was a cute picture with the piano playing and stuff.
Yeah,
So
my dogs like to listen.
hey, something. Sure, they can't applaud, but that's all right. They can do it. They can like it all they want. No, I've always been jealous of anybody that can play an instrument, because I just don't have the wherewithal to that. I can
Well,
play
you
a
need
mean
an
air guitar.
audience
That's
too.
about it. So
Yeah.
I can fake it really well. I did an air guitar contest once in a bar, and I won, and the judge was giving me the cash I had won, and he says, well, he said, it's really not fair, because it's pretty obvious what you're doing. And I held up my hands. I said, you put strings on these things, I'm done. I have no idea. I said, I can't even read music. He said, really? I said, no, never learned. So
I got to see you doing the air guitar when we meet in People's Pint.
I can do this. Oh my gosh. Yeah. So we'll get into a little semi-serious stuff. Now, where did you go to college? Because obviously you're a UPenn, now. But then how did you get from high school to UPenn?
I still am, perpetual student. I went to college in my hometown at Yale. My father was a professor there, and I was a faculty brat. And that's one reason why we still go to Western Massachusetts, is we built a house on weekends and summers. My father, being a physicist, was able to direct the winches with the wenches.
Nice.
And and then I moved on from there. I knew biology was what I wanted to do, and so I went to a graduate program, biology, and did some postdoctoral training, and then realized, hey, there's a lot of technology developing. This was in around 1980, to be able to manipulate genes around. And that would be the way to treat a disease at its root. We didn't know what many of the diseases were from a gene perspective at that time, but it was just really something which caught me. I wanted to do this. And so I applied to medical school at that point. Whereas in college, I was anti-medical school. I had a big button saying, I am not pre-med, because I couldn't stand that pre-med competition.
Nice.
And so I went to medical school to learn about the diseases and figure out what would be good targets for gene-based treatments. So back to your question, I went to school for decades.
I graduated School of Life. That's me. Somebody asked me once, he said, did you ever go to college? I said, I went to a lot of colleges. Never signed up for classes or anything, but I've been to a lot of colleges.
Well, so, I do believe in being a perpetual student. So many things to learn about.
There is, and really, I joke about it, but I'm one of those people. I like to learn. I like watching History Channel and National Geographic, things like that, and just learning stuff. It's interesting things. So now, So then that path led you to obviously choroideremia. Now, when did you first hear about that?
I first heard about choroideremia when Franz Kramers and his colleagues published a study linking the disease, showing there is a deletion. At that point in time, which was around 1989, it was really cumbersome to identify genes and figure out mutations and how they would cause disease. And really, choroideremia was the first one where there was a really good clue. And I actually met the person whose DNA was being studied in that paper, Frank Brunsman. I don't know if you ever met him.
Oh yeah,
from Germany, or Europe.
Yeah.
And so that really attracted my attention. This was before the Human Genome Project started, before it was possible to amplify and clone genes really easily. PCR machines were not commercially available. PCR machines were not available, and it was really around that time that they started to be produced and become available. So choroideremia was really number one.
Wow.
Of course, we didn't know very much about it. We didn't have the ingredients. I learned more about it actually right after I took my first job, which was at University of Pennsylvania. My husband and Albert McGuire and I were jointly recruited in 1992. And in early 1993, one of the low vision specialists called me and said, we have this 16-year-old boy in here who has RP, and he has a really large family. He was just telling me about his grandfather who had RP and his cousin and this, that, and the other thing. And he wants to do something about it. And I said, oh, great. Let's figure out what to do. And can I say his name?
Yeah, because he already talked about this when I interviewed him a few months ago. So
oh, awesome. Okay. So this person was none other than Chris Moen, age 16 when I met him. And we got together and figured out how to bring in his entire family, essentially, over a weekend. There were several retina specialists there who were doing evaluations, and everybody gave a blood sample. And we collected this blood, thinking this was a form of RP, brought it back to the lab. And I remember it actually coincided with Albert's in my wedding anniversary, and we were supposed to go out to dinner, and I was in the lab like purifying DNA. So like, okay, this will wait. I think we celebrated the following day. But we purified this DNA and started looking for linkage, which is what was being done at that point in time. We didn't have candidate genes to look at. The only candidate gene was rhodopsin at that point in time, and clearly it wasn't rhodopsin-based retinitis pigmentosa. And we joined forces with a non-Swarup and did linkage analysis, and lo and behold, it was choroideremia. So wrong diagnosis. We found out what the right diagnosis was for this family, which was really remarkable.
Probably the first person to be correctly diagnosed as a CHM using genetic testing, really, when you think about it.
Wow.
I mean, it's pretty common now. You'd be surprised. Yeah. We did a poll, and that's what brought about, I mentioned that differential diagnosis thing we did. We did a poll within our support group, which is, oh gosh, close to 1,500 people in our support group. And basically the question was, think about the first person in your family who was diagnosed. Was it choroideremia or was it RP? And 70% of the respondents was RP.
Wow.
So yeah, that much of an issue. So yeah, so Chris was the first one to get corrected. See that? But that's really, and that kind of led you along that path then to get more and more interested in choroideremia.
Oh yeah. Oh
because-
yeah. Well, like when I met Chris, his grandfather was blind at that point. I got to know the whole family, and the modus operandi became, we have to help Chris. We have to figure out what to do to
Mhm.
help him and his family. And that was 30 years ago.
Gosh, that was even before we had a support group.
Yes.
That started in 1996.
Yes.
The first
yes.
email-based support group. That's when I first contacted you.
Yeah. And I remember that so clearly. There were only a few people.
Yeah. Yeah, at that time. Yep. And we were only in touch with, well, let's see, Frank is kind of a CHMer slash, a doctor himself, but then there was you, there was Dr. Mack, and there was Dr. Kramer's.
Yeah.
And that was about the only people. And then Miguel came on
Right.
soon after that. Those were about the only people doing research at that time, like really looking at the nitty gritty of choroideremia at that point. You had a lot of other doctors like Dr. Burson and stuff who, they're following patients and doing studies and, and things like that. But, but that was the beginnings of the really, get down into the nitty gritty of things at that time. So, yeah,
and
I remember
what
those
the
days.
Choroideremia Research Foundation has done over the past few decades. It's just astounding, just miraculous. The energy and enthusiasm to support all the studies that have gone on, amazing.
Yeah, we've been I've said for years this would make a good movie. This little patient group, a few CHMers decided, hey, we don't want to wait around to go blind, and let's see what we can do. And we've come quite a long way in 22 years. So I'm looking forward to our celebration in June, up in Rochester. So, and on that, so now you've attended a few conferences. And what was, from a personal and professional perspective, what did you take away from those conferences?
I found those meetings motivating, not just from a scientific level, which, that was just extraordinary, having all the experts in the world right there to discuss things and share data, but also from a personal level to network with people around the world and to discuss issues they face and get help and ideas about how to move forward with clinical trials and testing and genotyping and cell models, animal models, everything. I think these meetings have been the perfect opportunity for the patients to talk directly to the people running the science, and they're interdependent. They're essential because neither group knows enough about what the other is doing. The sharing of information has been critical, I think, to move this field forward.
Yeah, and I think that, and maybe I'm wrong on this, but I've always felt that, when the researchers come to the conferences and they meet the CHMers, putting a face to their research, putting a face on their data, I think is a big plus.
There is nothing more motivating, I can tell you, as a researcher, to talk to people and see how difficult their life is if they're facing a disease like this. And I have tried throughout my career as a faculty member to make sure that my students get that experience of working with patients or talking to them or going to a meeting where their patients, because they come back energized and fueled and determined to make a difference.
Good. Yeah. And like I said, I think that's a very key thing. It's not just the money. It's not just the lab. It's like, there's patients behind this, you know? And of course, us patients, we have to have patience because research takes a long time.
I know. We all need patience.
But I remember reading an article, and I can't remember what year it was, but it had to have been early on in your research, but it was something you were had a hypothesis at that point of being able to use, this is before we had animal models and everything else, but being able to use skin cells as a proxy for retinal cells for doing research.
Yes, indeed. So you're right.
Remember when that was, but it was a while ago.
This was really early. I believe it was 1994. So remember, I told you that we first met this choroideremia family 1993. It wasn't long after that, but we were using there are a number of ingredients that are important for being able to make an effective gene therapy. And one of them is a way to deliver the corrective gene. And at that point in time, we had the first vector that was used to deliver genes called adenovirus. That turned out to have been very immunogenic and is not the ideal virus for delivering a gene which you want to be around long term. And however, it's all we had at that point in time. And we didn't have an animal model, we didn't have cell models, but Chris Moen and his grandfather and other members of his family came into the lab. And I remember like Al took skin biopsies and he did it from their butt so that if there was a scar, nobody would know. Drop your pants in the lab.
Oh my gosh.
And from those biopsies, we grew fibroblasts, they're skin cells that are grown in culture, and treated those and showed that we could indeed correct the enzymatic defect using a gene. And this was a collaboration also with Franz Kremers, because he and his colleagues sent their cloned choroideremia gene, which we then kind of optimized and put into this viral vector. So yeah, that was a long time ago.
Yeah. Yeah, that was, like I said, I remember seeing that and saying, so fast forwarding, how do you feel about the current state of research with choroideremia? And yes, we have come a long way, but obviously we still have a long way to go, both from the halting progression end of things, and also long term of being able to replace what we've lost.
Right. So the good and the bad. On the good news, I think, is big pharma and small pharma has been attracted to this disease, this ultra rare disease that nobody knew anything about 30 years ago, and they went into it full force. Several different companies, and there's a clinical trial going on right now with 4D, but starting with Spark Therapeutics and Nightstar, and then Biogen. Getting from this like proof of concept laboratory phase into the clinic takes an extraordinary amount of money and regulatory work and bench work and all sorts of things, which an academic lab can't do. So the fact that this became a target for these small and large companies and became like almost a household word, choroideremia is now known to investors on Wall Street and so forth. That's just fantastic. And I think the data from the trials that I've seen, for example, the Spark Therapeutics data and the Nightstar / Biogen trials, it shows a very high degree of safety. Yes, there have been a couple of problems, but with the AAV vector injected subretinally, it's really been extraordinarily safe. And when these studies started, everybody was so concerned about injecting under this teeny little area, which only has a few cells left. And they survived. That's really amazing. And the risk, I got to say, the risks that the choroideremia participants took to do that, that's heroic as well. Risking going from pretty good visual acuity to none, like that's you guys are heroes to show that this is safe.
Yeah, I think about that. Ooh.
Yeah, I know. And I think the data, we need to learn more about the data. None of the data sets from the large studies have been published yet. I know that the Spark data has been submitted as a manuscript and got good reviews, and so that will be out sometime soon, I predict. The Biogen data from phase 3, I really hope that will become available so that people can learn from it and figure out what the next step is. And of course, Robert McLaren's group from Nightstar X has published their earlier phase data, which has been really great. Of course, there are a couple of complications that have happened, and like for those particular individuals, that's not so great. But I think the vast majority, it's been great. Here's the bad news, in my view, is that a lot of people have said that these trials failed. They did not fail. I think let's start with the Spark study. That was a phase 1 study, phase 1/2. That means it was a safety study, of course, hoping for efficacy and selecting people based on risk benefit ratios, and basically looking to see whether this is safe and hopefully whether there's some efficacy and what evaluations to use if this goes forward. The Spark study enrolled 15 people. And these are they were followed at 2 different centers. That's an extraordinarily low number of patients to evaluate to look for efficacy. You can't look for efficacy. It's not statistically powered. What it did show is there was safety, and that safety was robust. And I think the next step, and it showed what some outcome measures could be for follow-up in phase 3 if this is to go forward. And it gave some ideas about how to proceed, I think. But sadly, Spark Therapeutics decided to stop after this phase 1, which implies that the study is a failure. It is not a failure, okay? And let's get back to how this could proceed in the future.
And I think, just to toss this in too, I think a lot of it came down to how are you defining efficacy? Are you defining efficacy as improvement, or are you defining efficacy as stabilization? And then how do you prove out either one of those?
That is a critical point. So gene therapy is not going to bring back cells that have already died, which is what has happened in the subjects who were enrolled in the Spark trial. They only had tiny little islands of vision. You're not going to bring back the rest of the photoreceptors. The goal here really was to maintain the vision that was already there. A lot of the individuals had pretty good visual acuity, and if that was maintained, that would be a meaningful outcome. And I really think that that the results were good in terms of that efficacy. Of course, you want to see better than that. You want to see improved visual acuity. The trial wasn't powered for that. It wasn't looking for that. And And then for the Nightstar/Biogen phase 3 trial, unfortunately, they chose an outcome measure, which would have been extraordinarily difficult to hit. 15 letters on an eye chart that's a really tough measure, particularly for choroideremia, where people tend to have pretty good visual acuity to begin with, and to hope that you're going to get a lot better than that is hard. And so it's sad that it didn't meet that outcome measure. But again, we don't know about the other data in the trial. Was that the success? Let me back up a little bit. The success of a trial, it's predestined. You have to say before the trial starts what your criteria for deeming it success would be, and so if they hit success on a secondary or exploratory outcome measure, that would not be counted as a success, where it really it should be. And so it will. That's one reason why it'll be really important to see all the data from that trial, because it may give an idea of how to move forward using alternative outcome measures in additional trials.
Yeah. And even if it's negative, as they say, even negatives can be a positive, because it can point you in a different direction.
It's research, and we have to learn what happens, and then get that feedback, and then figure out how to move forward.
Yeah, I remember, gosh, it was one of our get-togethers at our meetup place in Western Mass. But I was at that time under consideration for the phase 3, the Biogen phase 3, and I remember you saying to me, remember, this is experimental, it is research.
Mm-hmm.
And I said, yep, I fully understand that. And I also, and it was funny that the day before the surgery, I sat down with Dr. Panisi and Dr. Lauer, who did the actual surgery. And I was kind of chuckling to myself. They were going over every single bad thing that could go wrong.
Mm-hmm. Mm-hmm.
Up to and including you'll be under sedation, you could die. They threw that one in there too.
Sure.
But they got through the whole thing, and they said, are you willing to, move forward? And I said, here's the way I see this. If I do nothing, I already know what's going to happen in about 5 or 6 years.
Mm-hmm. Mm-hmm.
So, let's say worst comes to worst, I wake up, I'm blind in that eye. My timeline got advanced by about 5 or 6 years.
Mm-hmm.
So it's not something that was unexpected to begin with.
Right.
Right. This is what has been my fate since I was 4 years old. Mm-hmm. Mm-hmm. And so was it worth the risk? Right. Yeah, right. Absolutely. I mean, now that mindset for someone who's 25, it's going to be a little different.
Right.
I was 54 and, at 1 degree of periphery when I had my surgery. Mm-hmm. So there's a big difference there.
Sure. Well, so that you got to a point that I feel passionate about. And that is, okay, one of the challenges with this disease is it takes a while to progress. That's a good thing. Mm-hmm. For choroideremia patients. But it's a very difficult thing for pharma, because they need to see a result in a short period of time, ideally 2 years. 2 years, it's really hard to measure any change in choroideremia. 5 years, maybe you can measure change, and you just stated that. I would like to see a discussion, and maybe this is something the Choroideremia Research Foundation can start, about enrolling younger patients. And you just said, well, think about it. If you're 25, maybe you don't want to enroll in something like this. Okay. My hypothesis now is that we've shown this is safe. for example, the Spark vector and the Biogen Nightstar vector, which are very similar reagents. And we know from various laboratory studies, this works, at least in cells and in animal models. What can we do to start testing this in younger individuals who still have cells in the peripheral retina and who are undergoing degenerative changes at a fast rate so that an outcome measure can be determined in a relatively short period of time? Now, this is a real challenge because it involves ethics, and it involves enrolling younger people who are doing pretty well with their vision and haven't hit that scary point yet where they see it disappearing, but where it is disappearing, and we could potentially, we know the outcome measures to look at rescue of those peripheral cells. How do we deal with the ethics of enrolling somebody with good vision who could risk losing some of that in the periphery, perhaps, at a younger stage of the disease?
Yeah, that is a tough one as to how do you weigh that? You know, the technically risk versus reward factor.
Yeah. Yeah.
You know, and they're and like I said, for someone like myself, it's an easier question to answer. So
yeah. Could you enroll a 16-year-old? Like, think back to when you were 16. Would you have tried something like this? No, like having good vision at that time?
Yeah, I wouldn't have had time. I was competing in martial arts tournaments and, you know, playing volleyball, and I wouldn't have been able to take the time off at that point. No.
Yeah, that's a tough one.
Yeah, and especially at that age too. I mean, and not just, and I'm not talking just CHMers, any person at that age, they're bulletproof. At least they think they are.
Yeah.
You know, but that is the mindset of a teenager. You're, you know, heck, I'm not even sure if at, 21 or 22.
Right.
I would make that, even though by that point I was just about legally blind. But I don't know. I don't know if I would have.
Yeah. It's a tough one.
It is. It's a very tough question. So, well, listen, so do you have any last words that you would like to give to the CHM community in whole? And, I know a lot of them already, so.
The CHM community is like family to me. There is something that I think is linked to choroideremia, and that is the nice gene. It's just such a wonderful group, and so I can't thank the CRF enough for what they have done to advance this field, looking at every aspect, like the funding that you've provided for me including for things like equipment and sponsoring research projects that are experimental, looking at alternative methods to gene therapy, looking at cell biology and cell models and generating a choroideremia pig. I mean, it's just, it's phenomenal. You guys are on top of everything, and you do it with such great humor and enthusiasm and dedication. And so I would just say you are the role model for everybody. It's extraordinary. In fact, let me just put on. I wear this proudly. I don't do karate, but I do kickboxing.
Hey, close enough. That is awesome. Another member of the team CHM. Yeah, and it looks like I guess the $1 Million Bike Ride is going to happen in Philly again this year.
That's great.
At least at this point. So yeah, hopefully I'll try to make it down for that. Well, Dr. Bennett, I am so happy you were able to sit down with me today and have a little chat. And
I really enjoyed it.
Yeah, thank you very much. And hopefully we'll, like I said, we'll have to keep in touch and make plans, because, it's been a while.
Yeah. Looking
know?
forward to seeing you in person.
Yeah, absolutely. All right, Dr. Well, thank you again. And
Thank
we
you.
will hope to talk to you again soon.
And thank you so much. And thanks to Todd for putting the video together.
That's right. He can make even me look good. All right.
Okay.
Take care.
Okay.